the important topic like how to analyze the transcriptomic results. Like many of you are asking, that many of you are my friends and many of my juniors were asking like how you did a transcriptomics like analysis and what are the components. Actually, we need to take care while doing the RNA-Seq studies. Actually, it is also called as an RNA-Seq. How exactly the files look like, what to do with that files, and what analysis can we make, we will see in this lecture. First of all, I want to show like my research paper, like my research, like how I did my analysis, and we'll forward. Basic transcriptomic results we need to analyze. For example, if you send an RNA to the any outsourcing for sequencing, they will send you the file called as a multi-QC file, which includes means a quality check of the multiple sequences. Like you're doing a particular sample, you are doing a quality check means that is a QC report, but they compare across the sample, that is a multi-QC check. Before going to any further analysis or before going to any other transcriptomic analysis like RNA-seq pipeline, you should have you should must go with this actually. Say reports like I have sent in a samples like this nearly 8 samples I sent for sequencing, and it will done in paired end sequencing with Illumina. The base pair like read length was 150 base pairs. Importantly, like PB like I have sent and I have done it in 3 replications. You can see PBH 1, 2, 3. 3 technical replications I have made actually. This is how the data looks like how we have to code actually. We have to code very nicely. If your code is not good, like your label is not good, everything will mess up. Then you should look at the first 3 parameter is like how many number of percent of duplicates and how much percent of GC, and what is the read sequence, how many millions of reads we are getting. Like in my sample, I got nearly around average of 50% of duplications and 50% of GC. If this much is good, this is okay with, you can go with further analysis. But one sample I got, see, you can leave. There is only 46% of GC. I didn't get any reads in the sample, and I got 5.9% of duplicates. Okay, I have resent this sample for their analysis, and they have read it actually. This is how like you had to analyze your quality check report. For example, you check you sequence counts, like how many unique roots are there, how many duplicates reads are there. Like I told, like 40 to 50% duplicate reads above, then like below, then it's very good. Like a mean quality score, it's a very important thing you have to look at. Like a quality score, like per see average quality score, like it should be 30. Normally we select it for 30 across the position of the my genome. The all the bases passed a quality check. The sequence has to pass it to quality check. Means it's like that. If you are sequencing 1000 base, there is a 1 error. Means your quality check is like it's a 30. Sorry, the Fred score value is 30. If you are getting 1 error in if you are sequencing 100 base, but you are getting 1 error, means that is Fred score is 20. Normally, for in case of rice, we fix that like Fred score above 30 is very good. It has to pass this quality stringent check. Then like per sequence quality scores you need to check. You see, it's almost of my sequence is around like 30 to 46. Means it's a good Fred score is very good. Means if I analyzing a 1000 base, only 1 error is getting. Like this, we had to the Fred score above 30 only. The bases which having the above 30 only you have to sequence. Like per base sequence content, per base n content, you should look, and you should according to your species level, you have to see which either it is past their cutoff or not. Then after analyzing your sequence data, then you have to see the what is the protocol. Like this is my data actually. First, I will tell the protocol. First, we having a sequence data. Then we having a. Then you have to filter out your data. Like for example, as after quality check, based on that, why to filter for example, while sequencing, what we will do, we will add an adapters at the end of your reads. Like uh, every read having an adapters. We have to remove that adapters, and also based on the Fred score, sequence is having the below Fred score. The reads which having the Fred score below 30, we have to remove that. Like that, we have to filter out the reads. Then only after filter outing the process, we have to check the filter reads. Then those reads can be used for the alignment purpose with the reference genome. Then after alignment, we have to assemble your transcript. Okay, like after alignment, we will get an assembled sequence. Then after assembly, we have to count like what is the amount of transcript per exon, like that. After that, by using Dissect or any other software, we have to calculate the differential gene expression. If you want to know more, please join my course. Uh, then then we can go with a gene ontology analysis, like where we classify the our differentially expressed genes into different components like biological processes and molecular function, and also with the cellular components. Like this, we classify and the KEK pathways. Like this, after doing this is the overall pathway for transcriptomic analysis. Then, when you look into the data, where is my data? You will get files like this based on like I have sent. I sent 8 samples and I made 5 comparisons. Comparisons you can make. Either you can want to see resistant person susceptible, you can go with it. But only one thing you should consider: if you are resistant and susceptible having entirely a different background, my suggestion, don't compare them. Okay, because if the genotype is entirely different, you'll get a lot of background noise. Okay, if you are having a resistant and susceptible, like for example near resistant plaques, if nils are one nil is resistant, another nil is susceptible, they are only differ with the your resistant. At that time we can go with RNA-Seq comparing the susceptible nil and resistant nil, because almost your background is the same. They only differ with what are the genes you are searching for. But if you are comparing a resistant and susceptible, so much background noise will be there, then you can't go with anything. Then after that, how the results will look like. For example, first we have to check what is the like Excel sheets, how it look like. For example, this is the Excel sheet. Like first, there will be raw result. Actually, you can ask company to do whatever. Then either they can send reads of each sample, like transcript abundance of each sample. You can do dissect and you can generate this way. Either you can ask, better you ask the company people to send the DAG file itself. So for because of many of we are not bioinformaticians, we are breeders. We should not we don't need to learn these things actually. We just know how to analyze this stuff. Okay? We have to know how to the downstream end of this analysis, rather than doing how to do dissect, how to do check the transcript abundance. If you want to learn, please subscribe my channel. That is free join my course. Then like transcripts will be there. Then a locus will be there. Chromosome, which chromosome, start position, end position, your gene ID, protein ID. This is called as annotation. For every sequence, they were annotated. Okay. Then everything we have based on the p-value. Many people will like based on log2 file well maybe. No. Actually, it is based on the p-value. In the QRT-PCR, you may take log2 folds and well. But we having here a multiple comparisons. Means, for example, you are accepting a 0.0. That's why you have to take a p-adjusted value. Normally, we look into the p-value, but we're having a multiple comparison. That's why we have to look at p-adjusted value. I think many of you know that what is the correction factor, what is fault discovery rate, benzymanic correction factor. That things you should know. Okay. Then like either it's down regulated or up regulated. After that, annotation like geo annotation. Like there are several softwares for there to do this. If you want to learn, please subscribe my channel. Like if you want to do geo annotation, like there is a software called David. Either you can use TB tools. There are many other n number. Either you can go Agrigo. There are several softwares are there. You can do Jiva ontology. It's not like that company people will do. You can easily, by one click, if you submit your DGs, it will give the Jiva annotation. Like this, it will give Jiva annotation, like cellular components, biological process, molecular function, tech pathway. I am interested only. There are several methods, several others also there, but I am interested in this. Then, like see here also we are not getting anything. So there are so many pathways are there. How can I narrow down my genes of interest? You can go with based on a further p-value which is significant. You can check top enriched pathways. Like see in my case, the top enriched pathway actually this is a susceptible. Uh, 33 genes were associated defense and rest other with the photosynthesis. Then these are a simple metabolic pathways, photosynthesis, synphotor activity. Based on that, you should think that which genotype is chosen and what are the possible mechanisms. For in my case, it's a nematode interaction. I should know like when the nematode infect the genotype, what will happen actually. If you don't have any information, what are the result you got itself you publish. After infection, highly enriched pathways are different and here count is 3 and what are the genes. Then you have to compare these genes with the annotation what are the because you have annotation table. Simple VLOOKUP formula you have to use to check which genotype which annotated with which protein. Then you will design this is then you will design which pathways are highly responsible and highly differentially expressed when your particular treatment was given. Like this, you have to analyze your data. Then there will be a plots. For example, you have to generate a plots like this. You can generate a plot based on the Excel sheet last I have shown. There will be like see you can see like defense response with the highly and cytoplasmic membrane was highly differentiated. These were the pathways are highly enriched when I am giving a particular treatment, so that I can generate some viewpoint. Like then you have to select the key genes. For example, already know that when the blast will affect, I know that already when nematode will affect, there will be a stubin there is suberin biosynthesis pathway is highly enriched. That is like the secondary metabolite pathways were highly enriched. I will look into the secondary metabolites which genes are there, what level of expression. Then I will pick some candidate genes and I will try to validate them. This is it's not that much complex. Not only this analysis, several analysis also we can do. I will show another analysis what I have done. Yeah, here you can see that. First you have to compare how many diseases were common between susceptible bulk versus susceptible infected, and I will show the one day when first you have to compare overall genes. Then yeah, I will show you actually. And please don't take screenshots and unpublished data. See, first we have to compare your disease. Like for example, I given a 4 samples. Like I have compared the disease with uninfected, infected. Then you have to overall comparison. Like 593 are exclusively for Pulerada and 83 exclusively for RB and 85, 859 for this susceptible bulk. Like this, you have to compare all the samples. Okay. Then you have to see the volcano plots. Like how much this will show that how your disease were there. For example, see up to minus I see 15, my genes were down regulated. Up to 12 upregulated, and these many genes were in the base. Like that, you have to check how the pattern of your expression of your genes in the sample when the particular treatment. Then I had told you how you can generate a gene ontology plots like this. Then you can compare it depends on you how you planned your experiment. I planned in a different way because I don't want to tell because it's unpublished. Then cellular response view this is a mapman analysis. If you want to know how to do this, please subscribe my channel. Like cellular response overview. For example, there is a one software called as mapman. If you put your digits, it will give like this diagrams. For example, I am done it for biotic stress pathways like ethylene pathways, oxygen path, like hormone signaling was highly enriched. based on like is blue is down regulated, red is up regulated. Like this, we can visualize what are the pathways are highly differential expressed when the particular treatment is there. This is what the beauty of RNA-seq. You will get a snapshot of genome-wide expression at particular treatment. This is exactly a beauty of like the metabolic profile. You can see, like a secondary metabolic profile. You can see this in this platform. Then you have to validate your results with qRT-PCR. Okay, this is what I want to show. It's in a quick way. This is what we have to do in detail. If you want to learn, please I have given a Google form link in my channel. Please join my course. Thank you.